So welcome everyone to the Diatom Web Academy. Let us know your name and where you're coming from in the chat. We welcome your questions in the chat box. We'll be monitoring that. And today we'll have time for questions in the middle of the presentation as well. This webinar series is brought to you by the Diatom Taxonomic Certification Committee, a group working to improve taxonomic data so that those data can be applied to research and management of aquatic ecosystems like that you will hear about today. I also want to bring to your attention that we are seeking nominations for topics and speakers to help us plan the webinar schedule for the coming months. So please visit the main Diatom Web Academy page to access the Google form where you can submit your feedback. So now I will invite Jan Stevenson to introduce today's speakers.
Hi, I'm Jan Stevenson. I'm one of the people on the Taxonomic Certification Committee. And it's my pleasure today to introduce Susie Theroux and Raphael Mazor from SCORP, the California Coastal Water Research Program. It's a group in Southern California that does a lot of the research and foundation work for the state of California to develop research programs. And I've been fortunate enough to serve on the Science Advisory Board and know these guys and really the cutting edge work that they're doing. So I thought they'd be a really great group to bring on board for one of the web academies. So with that, Susie got her PhD from Brown a number of years ago and joined SCORP and is now a senior scientist. And Raph, got his degree, PhD from the University of California a number of years ago, the University of California, Berkeley, and is as a principal scientist at SCORP. So with that, I'd like to just mention that the presentation is basically over California Algal Assessment. The first part of the presentation will cover morphological and kind of traditional taxonomic approaches and how that data is being used in the assessment program for the state of California. And the latter part will be about molecular methods. So to give a little break in the presentation, which will probably go about 50 minutes, we're going to stop for about 5 minutes in the middle of it before we start the molecular part to see if there are any questions. So with that, I'll turn it over to Susie and Rafi. Thank you.
Great, thank you, Jan. Thank you, Sylvia, and thanks so much, everyone, for joining today. I'm really excited to be part of the Diatom Web Academy. I've really enjoyed some of the previous presentations, so I think this is going to be a lot of fun. My job today is to give you an overview of the California Algal Bioassessment Program, and I'll walk through some of the indicator and tool development that we've been doing and some of the potential applications of the tools that we're developing and really where we see this program going in the future. Thankfully, we have Raphael Mazor with us today as well, and he's really working on some of the implementation elements of the bioassessment tools that we're developing. So when those sorts of questions come up, it's great that we'll have him online as well. So I wanted to provide a little bit of a big picture overview of how we think about our California Algal Assessment Program. And I was thinking back in the creation of this PowerPoint about when I first moved to California, and I knew that I was going to love being in California because of the wonderful weather and the sunny beaches. But little did I know I'd also fall in love with California because of its wonderful algae program. And so what I think really lends to the strength of the California Algal program is that it's focused on taking biological data and turning it into action. And so what do I mean by that? So there are kind of three main pillars, I would say, to how we think about turning biological data into implementation and actionable information. And that's first off, making sure that the data that we're collecting is really robust, that it's collected using sound scientific techniques. and that we're using this biological data to develop strong analytical tools that give us precise and accurate biological assessments. And that also then we can turn all of that biological data into information that can be easily interpreted and readily communicated to our stakeholder communities for making regulatory and management decisions. And so we're always keeping that in the back of our mind as we're developing these tools. We want to make sure that they're based in sound science, and that's going to help us have better bioassessment outcomes. So as Jan mentioned, Rafi and I come from an organization called SCWRP, and I thought it would be useful to provide a little bit of background about how we came about to be the ones doing a lot of the development of some of these bioassessment tools in California. So SCWRP is the Southern California Coastal Water Research Project, and as our tagline says, we are interested in applying these next generation science to aquatic ecosystems management. So what we are is a public agency, and we're a joint powers authority. And so we have a 14-member commission that is comprised of both regulatory and regulated entities. And so from our regulatory side, we have representatives from the State Water Resources Control Board, US EPA, Region 9, where we are, also the Ocean Protection Council. And then for some of our regulated members, we have representatives from wastewater and stormwater agencies all throughout Southern California. And so we work at the interface of both the regulatory and the regulated entities, focused on being a non-biased science research organization that's developing tools that's going to benefit all of our member agencies and the state at large. I like to show these two pictures when I'm explaining exactly what SCWRP does, because I think it really represents kind of the different kinds of hats that we wear as SCWRP scientists. So sometimes we're out in the field, we're collecting biological data, and then a big component of our work is to then translate that biological data into actionable information, and so sharing what we've learned and the tools that we've developed with the stakeholder communities. All right, so why are we so interested in generating bioassessment data or using bioassessment? I think that this slide will provide you a little bit of the regulatory context of why we like to do bioassessment. The Clean Water Act compels us to restore and maintain the chemical, physical, and biological integrity of the nation's waters. And so in 1997, the strategic plan for the California Water Resources Control Board formed this bioassessment monitoring program, and that really launched the bioassessment program as we know it today. Why do we like doing bioassessment? I think I'm really preaching to the choir if you've joined this webinar, but as a reminder, there's really been a global paradigm shift towards using biological data. It allows us to have direct measures of the biological communities that we're interested in protecting. And also bioassessment is a tool that is integrative over time. So as opposed to a snapshot of what chemical composition might be like in the water, biology that's living in the environment is integrating all of those impacts over time and really helps us focus on our ultimate policy goals of protecting the biological integrity. In California, what are some of the current and anticipated uses of bioassessment? We have a number of different ways that we're turning that biological data into actionable information, and largely we see it in the form of baseline and trend monitoring. We evaluate the effectiveness of things like best management practice implementation, restoration projects, permit requirements, remediation efforts, and also working towards establishing biological targets for management goals, and also helping us evaluate reference conditions. So when I'm talking about the biological data, I'm largely referring to right now our use of benthic macroinvertebrate data for performing a number of these activities. And we have the anticipated use of the algal data also being able to fulfill a number of these roles. The figure that you see on the right-hand side of your screen is showing how we're using that benthic macroinvertebrate data for integration into our integrated reports, which are part of our EPA requirements, and identifying our category 1, so our pretty pristine versus our category 4 or 5, our impacted stream environments in Southern California. So in California, our wadeable stream biomonitoring has really been the pillar of our bioassessment program. And so we can credit that to something called the Surface Water Ambient Monitoring Program. And so this is the bioassessment arm here in California that's really responsible for all of the great bioassessment monitoring and data collection that's happening all throughout our wadeable stream environments. So there are two main programs within the Surface Water Ambient Monitoring Program. One that's called the Perennial Stream Assessment, or the PSA, and one that's called the Reference Condition Monitoring Program, or the RCMP. And so the PSA is responsible for collecting biological data in addition to physical habitat data and vegetation data. And it uses a randomized stratified sampling design to target as many sites as possible all throughout the state. In contrast, the Reference Condition Monitoring Program, as the name suggests, is specifically focused on those reference sites, so those ones that we believe are representative of fairly unimpacted condition. So thankfully, because of the strength of the SWAMP program, we have been collecting over hundreds of sites every year for over 10 years, which has resulted in thousands of sites that have had this data collected that we can now turn into useful biological indices, bioindicators, and eventually actionable information for management decisions. So one of the first ways that we began to really capitalize on this excellent, robust biological data that's getting collected in all these different stream environments was in the creation of the California Stream Condition Index. So the California Stream Condition Index is a benthic macroinvertebrate biological index. And as I mentioned, it was built on the foundation of that biological data that was collected through the SWAMP program, but also in addition to a number of regional monitoring programs that are using the same standardized sampling protocols as have been outlined in the SWAMP program. And what's special about the CSCI is that it is a predictive index. And so what I mean by predictive index is that when we're evaluating biological condition all across the state, we don't have one expectation for all sites and how they're performing. We have all tailor-made expectations depending on where those sites are situated and their geographical setting. So in a predictive index, you essentially incorporate what we call site-specific or reference-based expectations for your species distributions and the ecological structure of your biological communities. This makes it a really powerful index that can be applied statewide, and it's currently being used for regulatory decisions and the development of biocriteria in California. So this index has been really successful and has been shown to be applicable in many different environments all across the state. But now we want a similar complementary index for algae. We've seen that the CSCI, or this bug-based biological index, has been so successful, widely adopted, and now we want the same thing for our algal communities. One of the main reasons that we want to now have a second biological index is that we know algae respond to different stressors from the benthic macroinvertebrates. We know that they're really important in biogeochemical cycling. They're sensitive indicators for nutrients. They respond really rapidly to change. And they're also highly diverse and ubiquitous. We find them throughout these stream environments. So again, we can rely on the foundation of that strong biological data that's been collected through our statewide and regional monitoring programs to begin to develop a novel index based on algae. For a little bit of background on what the algae sampling actually looks like in these stream environments, as I mentioned, one of the real strengths of the program is that we use these standardized protocols. So the snapshot that you see on the right-hand side of your screen is our standardized protocol for that includes the collection of our benthic algae in our wadeable stream environments. And so our protocol involves sampling multiple transects along 150-meter reach in the stream, and it's a multi-habitat collection method. So we're targeting all different substrates, and then we're compositing our algal communities into one final composite sample that then we divide up for analyses in the lab. One of the things that is unique about how we're doing algae taxonomy for stream samples in California is that we look at both our diatom and our soft-bodied algae communities. So we take that composite sample, we divide it up into the microscopy fraction that's going to be looked at for diatoms and soft algae. And then we also look at the soft algae macro algal fraction. And so we get relative biomass and relative biovolume estimates for all of our diatoms and soft-bodied algae. And our soft-bodied algae also includes our cyanobacterial populations. So this is a lot of work, but it results in really great taxonomic data that we can use for building our biological indices and our bioassessment tools. So first off, in 2014, we completed the creation of the first Algal Index that we have here in California. We called this one the Southern California Index of Biotic Integrity. And as the name suggests, it was targeted mainly for use in Southern California. For the development of this index, we did use sites that had been sampled all throughout the state, but it was really calibrated to be used specifically in Southern California, where we had a really high density of sampling and therefore sites that were available for evaluating. For the development of this index, we had about 270 reference sites, which was good for calibrating our index. And because, as I mentioned, we look at both the diatoms and the soft-bodied algae populations, we explored a couple of different permutations of this biological index. So that included one that was focused just on diatoms, one that was focused just on soft-bodied algae, and then one that was actually a hybrid of the two. So we were looking at 3 different assemblages, and we explored something called a multimetric index. And so this is an index that really looks at the ecological structure of your biological communities that are present in all of your samples. For example, we might be looking at metrics that give us an evaluation of how sensitive or tolerant different taxa are, if they like high dissolved organic carbon, or if they're sensitive to copper, and all these different metrics get combined into a multimetric index. This was what we might call a traditional multimetric index, in that we had one expectation of reference condition for all of the sites that were being evaluated. And at the end of the day, this index was successful in developing 3 different versions of these multimetric indices. So we had a high-performing multimetric index that was focused on diatom populations, one that was focused on soft algae, and then one that was focused on the hybrid approach, where we combined both the diatom and the soft algae populations. The figures that I'm showing you here on the y-axis have the IBI score, so essentially the index score for all of these different multimetric indices. And on the x-axis, we see if we were evaluating sites that were considered heavily impacted or disturbed, our intermediate sites, and then also our reference sites. So as you might expect, reference sites tended to score higher. And as we can see, for each of these 3 different permutations of these multimetric indices, we had really nice discrimination in how our reference sites versus our heavily impacted sites were performing. So this was a really great starting point for us. We saw that algae were able to be used for index development in California. They were sensitive, they were responsive, they were helping us to discriminate from reference and impacted condition. And now we were greedy. We wanted to turn that into a statewide index that we could be applying at multiple locations all throughout California's diverse ecoregions. So that led us to develop a statewide algal index. This time we expanded the site pool that we're using for our development data set, no longer focused on just Southern California, looking at over 2,000 sites that were scattered all across the state. We had over 500 reference sites that we were using to calibrate our index. And again, we took the approach of using all of our different algal communities that we had taxonomic data for, stretching back over 10 years. So again, we had 3 different assemblages that we were evaluating in developing this biological index. We focused on diatoms, soft algae, the hybrid approach again. But the thing that was new in developing our statewide algal index was we wanted to see if we could incorporate a measure of prediction. So again, like the California Stream Condition Index that's based on benthic macroinvertebrates, we wanted to be able to predict a reference expectation that was specific to each of our individual test sites. To do this, we needed a lot of complementary metadata for all of our sites that we were sampling, and that included a lot of details about geographic setting, altitude, climate, precipitation, bedrock geology, all of these different components that we could use to predict our reference-based expectations of algal performance at all of these sites. Another thing that was unique in how we were developing this new statewide Algal Index was we were looking at two different types of indices. We were looking at an observed versus expected index, and then another type of multimetric index like we saw in the Southern California Index of Biotic Integrity. So the observed versus expected index, I know that you've covered this a little bit previously in a Diatom Web Academy seminar before, but essentially we consider observed versus expected indices a measure of taxonomic completeness. So we look at our individual test sites and we say, based on the geographic setting, what species do I expect to be present? And then you compare that to what species were actually observed. So as you can just see here, we might expect to see 5 different taxa present at a site. If you only observe 3, you are scored based on the deviation from that reference expectation. As I mentioned with the multimetric index, we're evaluating what we might call ecological structure. So all the different characteristics of what these different algae are doing in terms of their metabolisms or their habitats or what they're sensitive or tolerant to. So I'll dive in a little bit more in how we go about developing each of these different components of this statewide biological index. So that means we were setting ourselves up when we were developing this index for a number of different permutations. First off, we were looking at the comparison of how our diatom populations might do alone, how our soft-bodied algae taxonomy data might do alone, or what happens when we combine both our diatom and our soft-bodied algae data. Similarly, we wanted to see how did we do developing an O/E index? How did we do developing an MMI index? And could we have some version where we combine the performance of both, which is actually how the CSEI or that bug-based index is constructed. The other permutations that we were looking at was, should we be developing a regular multimetric index where we have one expectation for performance of the index across the state? Or do we want to incorporate this predictive element where we're tailoring our predictions based on the geographic setting of all of our different sampling locations? If you were curious how we went about deciding which of our sites we were going to use for calibrating our index, I just include this here as a reference. This applies statewide throughout California, how we're defining and identifying which of the sites we consider to be minimally impacted. So it's a screening criteria where we look at the environmental setting of these different sampling locations, and we identify using a number of GIS parameters and some field-based parameters as well. What's the percent of agriculture or urban development or our road density and our road crossings, and we set a number of thresholds. And if you pass those screening thresholds, you're included in our reference site pool. You get to be used for calibrating our index. If you exceed some of our thresholds, you're considered impacted or heavily impaired. And we use those sites for evaluating the performance of the index, but not calibrating the index per se. All right, so when you want to go about designing a predictive observed versus expected index, these are some of the steps that you could take. You start off by clustering all of your algal assemblages that you observe at your reference sites across the state. So as I mentioned, we had over 2,000 sites that we sampled. About 500 of those were our reference sites. We took our reference sites, we looked at the taxonomic composition of the diatoms and the soft-bodied algae and the hybrid, and we clustered all of these individual sites based on their similarity to each other. One thing that I'll mention when we were doing this for our index here in California is we actually converted all of our taxonomic data, which previously had been either count data, valve count, or it had been relative biovolume data. We converted all of it into presence absence. And I can talk about a couple of reasons why we chose to do that later, but it did help simplify some of our analyses down the road. So you start off by clustering your algal assemblages at ref sites across the state, and then you go about building predictive models using the geographic information that you have for all of your different sites, and you begin to be able to predict what species you expect to find in each of your different locations based on the geographic setting. In this way, you're developing these site-specific reference-based expectations of species distributions. What are some of the geographic parameters that we might be using for making these predictions? I've listed just a couple of them here as an example. We might use elevation, watershed area, catchment mean precipitation, local temperature, soil erodibility, soil permeability, and then some mineralogy characteristics that we've assembled for all of these different sampling sites. The thing that's important to note is that all these parameters that we're evaluating to help us potentially predict the species distributions across the state, they're all ones that we've decided are not influenced by human activity. So they're really hopefully tailoring our expectations, not to what the human impact should be doing, but what we should be expecting under pristine, unimpacted conditions. All right, so then we go about scoring our individual sites based on the deviation from the predicted values. So this graph here is just showing you your observed versus expected valuations of a number of different test sites. And you can see that the dotted line represents a one-to-one relationship where if all of your test sites fell on that line, they'd all be scoring perfectly. You'd be meeting your expectation based on your site-specific reference models that you have developed. But instead, what you normally see is a scatter because, of course, some of our sites, we're not observing all of the taxa that our models informed us we should be expecting given the sampling location. We follow a really similar process for developing predictive multimetric indices. This time, we start by calculating all of our candidate metrics. So I'm showing you a graph here of just an example of what it might look like for a metric such as % most tolerant taxa. So these are taxa that we know are pretty hardy, and so we expect them to be more abundant when you're at more impacted sites and in lower abundances at our reference or pristine sites. And so we calculate a number of candidate metrics for all of our different sites that we've sampled. And again, we build these predictive models to develop site-specific reference-based expectations of our metric performance for all of our different test sites. And then again, we're scoring our individual test sites based on the deviation from our predicted values. So here on the y-axis, we have what our observed values might look like for something like the percentage of most tolerant taxa present in our sample. And the dotted line represents the one-to-one relationship, where if you fall on this dotted line, you've met your expectation, you'll have a good metric score. If you deviate from that expectation, your score is going to be impacted, you'll have a lower score because you're no longer meeting that reference-based expectation. So what are some of the examples of the candidate algal metrics that we evaluated? There are a lot of the, there might be some familiar faces, I guess I would say. So they're fairly popular metrics that we harvested from a number of different online or published resources previously. We did some evaluations specifically for California, but I've listed just an example of what some of them are here. We tended to categorize them so you can see that there's a class of the type of metric that was included, if it was a measure of tolerance or motility, if it was a measure of an organism like certain requirements of dissolved oxygen or salinity preferences. We did use some measures of diversity, richness estimates, and then also we looked at taxonomic groupings for some key taxonomic groups that we know might be sensitive or tolerant species assemblages. Again, I'm just showing you an example here on the right-hand side of your screen of what the performance of some of those potential metrics can look like. Here we have an example of your % most tolerant taxa. So again, they're really hardy organisms, so they're increasing in percentages as you get into more stressed environments. And that's in comparison to some of your sensitive taxa, which you would expect to see those metrics decreasing as you get into more stressful environments. As I mentioned, we tend to assemble all of these candidate metrics from a number of different resources. We dive through the literature. We include observations from field and lab studies, so some empirical analyses. We did specific analyses for California taxa, so that was a series of indicator species analyses that we did to identify California-specific evaluations of sensitivities to nitrogen and phosphorus or indicators of high or low dissolved organic carbon. And we also sourced some of our potential metrics from other indices that have been developed, largely diatom indices that have been developed for some European programs. And we looked at what taxa were included in those indices as well and tested those as candidate metrics for the creation of our Algal Index. In total, we had about 150 candidate metrics that we evaluated for each our diatom, soft-bodied algae, and hybrid assemblages. And when we were evaluating each of those assemblages, on average, about 20 to 30 would pass some of our metric performance screens. So we were looking at things like, was it a sensitive index? Was it responsive to stressor gradients? Was it accurately predicting reference site populations? Was it consistent when we sampled across multiple samplings at a single sampling site multiple years? So we had a number of screening criteria for evaluating the metric performance. And when we took those between 20 to 30 metrics that passed that screening criteria, we then did the creation of what we call proto-MMIs. We did every possible combination of all of those different candidate metrics. We wanted to see if you took between 4 and 10 of those candidate metrics and you randomized which ones were included and you created these prototype MMIs, which ones ended up being the winner. When you do that, as you can imagine, your number of proto-MMIs rapidly inflates. So we would often have over 200,000 of these proto-MMIs that then we were needing some way to sort through to determine which of these indices ended up being the highest performing. So I wanted to get into how we go about evaluating index performance. So we have 3 key things that we're looking for when we're deciding if an index is passing our performance criteria. The first thing that we're looking for is accuracy. So we want to see that the index has no evidence of geographic influence. So as I mentioned, we're sampling sites all throughout California's diverse ecoregions, which I have a picture of here on the left-hand side of your screen. We want to make sure that there's not an artifact of the index performance based on where those test sites are. So the way that we do that is we look across our different ecoregions and we make sure that for our reference sites, they're all scoring as close to 1 as possible, which is where we calibrate our index. And then we're not seeing big shifts in the index performance at those reference sites based on the different ecoregions where we're sourcing our data. So the graph I'm showing you here is for an example of one of our multimetric indices. You can see that there's really nice consistency. On average, our reference site scores are scoring close to 1, and we don't see large biases based on the geographical setting for those different sampling sites. And the second thing that we're looking for is a measure of precision. So we want to make sure that we see really low variability in our reference site scores and also low variability in repeat samplings at individual sites across multiple years. So to visualize what it looks like when you have low variability in reference site scores, you're essentially looking for that box bounding the reference site scores that you can see here in this figure to be as small as possible. We really want some nice, precise, the reference site scores are as close to 1 as possible. And as I mentioned, we also want low variability in repeat visits. So when we're comparing how a site is scoring, if it's sampled over multiple years, that there's not large variability in the performance of that index. Lastly, we're looking at responsiveness. So we really want to see that this index is sensitive to stressor gradients. So some of the ways that we do that is by looking at how an index is scoring, as you can see here along the x-axis, in comparison to something like a known stressor, which is, for example, phosphorus concentration in the water of the streams that we're sampling. You can see in this example, we have a really nice responsive index where we're seeing our index scores are lowering when we're having our higher amounts of phosphorus. And that's what we want to see to ensure that we have a responsive, sensitive index. One of the other ways that we were evaluating index performance, and this was extremely helpful, was that we relied on an expert science panel to also help us derive our screening criteria for index performance. So you might recognize some of the faces that you see here, and they were all really instrumental in helping us establish robust screening criteria for the performance of our indices. So as I mentioned, we had 6 different permutations of these indices that we were looking at for our 3 different assemblages and comparing both the observed versus expected and then the predictive multimetric indices. So on the left-hand side of your screen, I'm showing you some of these box plots again, where we're comparing how our indices performed if they were being calculated for sites that we consider to be representative of reference condition or of high activity sites or of intermediate condition. And you can see that in these high performing indices, we expect to see this nice discrimination between our reference, intermediate, and stressed sites. And as you look across these 6 different permutations of the indices that we tested, there's some variability in the performance. There's some clear winners and losers. And so what I'll highlight for you here is that we essentially had 2 out of these 6 indices that we evaluated that really were the clear winners and had really strong, responsive, precise, and accurate performance. Those 2 clear winners were our diatom-only predictive multimetric index and what we call our hybrid multimetric index that included both the diatoms and the soft-bodied algal communities. So on the right-hand side of your screen, I'm showing you just an example of what the responsiveness looks like for those 2, or for all of our MMIs, but highlighting the 2 that were our high-performing ones, where you can see that there's just really nice responsiveness to a number of different stressor gradients, including here your percentage of urban development in your watershed, and some nutrient characteristics, the nitrogen and phosphorus in your water. You can see that consistently there's a decreasing scores when we're expecting like a highly impacted environment. So this summarizes the performance of these 6 different indices that we looked at. As I mentioned, the 2 real winners were the predictive multimetric indices for the diatom and the hybrid assemblages that we evaluated. And overall, our observed versus expected indices just had really weak performance. The diatom O/E index was borderline, but it really wasn't nearly as sensitive or responsive as the predictive multimetric indices that we developed. So we didn't consider them useful, and they weren't adding to the performance of our index. So our final indices that we decided on were the predictive diatom and hybrid multimetric indices. If you're curious what some of the metrics were that ended up in those final multimetric indices, I've listed them here for you. It's a bit of a laundry list, but you can see that it is a diversity of different types of these metrics that we included. In the creation of these indices, we made sure to not have too many metrics included that were duplicative or too similar. So you wouldn't want to have multiple metrics that were all evaluating percent or proportion of tolerant taxa. Instead, we wanted a diversity to give us a real holistic assessment as it got incorporated into the multimetric index. So you can see we have our diatom multimetric index, our soft-bodied algal, and our hybrid multimetric indices are all listed for you here to explore if you're interested. And I've also included here the different predictor variables that dictated the reference-based expectations for these different metrics and their performance. You can see that there's some consistency in the different predictors that were included. Some of the most popular ones were predicted background conductivity. So this is one of the metrics that actually uses a model to predict background conductivity, since we know that that is something that's influenced by human activity. We also see measures of soil erodibility and mean precipitation for a watershed. Those are all pretty popular predictor metrics that got selected in the creation of those models that we use for developing our reference expectations for metric performance. You might notice that for our soft-bodied algal multimetric index, which you can see listed on the bottom of the screen, none of those metrics ended up actually being predictive. And in general, as I mentioned, that multimetric index had fairly weak performance. So it just hasn't been recommended for future use, but I've included it here if you're curious to see what some of the different metrics are that get included in that multimetric index. All right, so now it really begs the logical question of why did we see such differences in the index performance with these different biological indices? And so I wanted to go in a little bit into why we see, or why we're suspecting that we see such differences in performance of our multimetric indices. So one of the things that we noticed when we were comparing our diatom and our soft-bodied algal communities is that there were pretty surprising differences between species richness and species similarity between our diatom and our soft-bodied algal communities at reference sites. So when we're looking at our diatom communities at reference sites, we saw on average higher species richness and greater overlap among the diatom communities than we did with our soft-bodied algae populations. So the figures here that you have on your right-hand side of the screen are some histogram plots that show you on average the Bray-Curtis distances. So the degree of overlap between the communities whether it was the diatoms, as you see shown here on the top, or the soft-bodied algal populations. We can see that Bray-Curtis distances were higher for our soft-bodied algae populations, meaning there was less and less overlap among the reference site populations. And then similarly with species richness, we can see total richness across all reference sites for diatoms and soft-bodied algae is actually fairly similar. We have about 700 total taxa for diatoms that we identify at reference sites. It's about 570 for soft-bodied algae, so not so different. But when we look at average species richness per site, we have almost twice as many diatom taxa on average that we're identifying at our reference sites in comparison to our soft-bodied algae. So how does this manifest itself in the performance of an index? Well, essentially, if we have much lower species richness at our sites, the expected taxa that we expect to see in our observed versus expected index performance just ends up being really, really low. So we were seeing numbers on average close to 5 taxa that were expected at a number of our sites. So when you have numbers that are that low, your models just get very unstable and you can't make accurate predictions. So in general, for our soft-bodied algae ovary indices, as well as the performance of these metrics, we just saw a lack of precision and a lack of responsiveness in these indices. And we really credit a lot of that to the fact that we had much lower species richness and a slightly smaller species overlap in our reference site pools. So the models just really struggled to be able to accurately predict species distributions and species ecological structure. If you're curious how this compares to benthic macroinvertebrates, you can see that even compared to both of our algal assemblages, the benthic macroinvertebrates on average are seeing species richnesses closer to about 56, which is more than twice the number of diatom taxa that we're seeing. So in general, this isn't something that we've seen plaguing the benthic macroinvertebrate communities to quite the same degree. It's a particular challenge for our data set with evaluating the algal populations, but we did see, and we credit a lot of the better performance of the diatom indices to the fact that they had slightly higher species richness numbers than our soft-bodied algal populations. When we think of our statewide programs, as I mentioned previously, we had this great example set forth for us of the benthic macroinvertebrate index, the California Stream Condition Index. And so as I mentioned, we were interested in initially developing this algal index as a second line of evidence or a complementary indicator to the CSCI, or that bug-based index. And so these two graphs here help to illustrate how there's some similarities and some important differences in how our now resulting algal indices are performing in comparison to the CSCI. So on the left-hand side of your screen, I have some examples of just some generalized additive models where we're comparing different stressor gradients on your x-axis to index scores on your y-axis. And each of these different lines represents the different assemblages and the different multimetric indices that we developed for our algal communities. And in purple, you can see the lines that are the California Stream Condition Index scores. So across what you see is that we're trending in similar directions. These indices are responsive to these different environmental stressors, but that there are some key differences where the lines are not matching exactly. We intend to see that the bug-based communities respond stronger to physical habitat changes. Our bug-based community, or our algal communities, respond slightly stronger in some instances to our nutrient enrichments. And so there are examples where we know the algal populations and the bug-based populations are responding to different stressors, and that's manifesting themselves in different performance of these indices. On the right-hand side of your screen, you can see again comparison where our hybrid multimetric index versus the California Stream Condition Index, where we essentially classified our different test sites as either passing or failing these indices. And so if it's colored blue, that means that at least one of the indices failed. And if it's colored green, that means at least one of the indices passed. But what's notable here is where we have a differential where either the algal-based index passed and the bug-based index failed, or vice versa. And so there are key locations here where the combination of both the algal and the benthic macroinvertebrate indices is giving us a more holistic, comprehensive assessment because we know that these populations are responding to different environmental stressors and on different time scales. So just to wrap up on the development of these algal biological index for the state of California, we now call it the Algal Stream Condition Index, or the ASCII. But really we have two different versions of these indices that we're now moving forward with. And so we have the diatom Predictive Multimetric Index, as I mentioned, and then also the hybrid Predictive Multimetric Index. This figure here on the right shows you some hybrid multimetric index scores all throughout the state of California. When you're in highly urbanized areas, as you might expect, you have lower scores. In some of our more remote, high mountain areas, we have higher algal index scores. These types of graphs are always good gut check to see our indices are performing as we expect them to. And now it's ready for prime time. So it's ready for application and implementation into a number of our statewide monitoring programs and for performing bioassessments and generating manageable, actionable decisions. If you're interested to learn more about how we develop this California Stream Condition Index, and you can access our development data sets and some of the trait attribute information that we use for developing this index and calculating our index scores, that is all available on this website. So I included it here if you want to learn more. It also links you to our GitHub where you can calculate these algal indices for yourself, which is a great way to begin to test drive this index in a number of different user community hands. So I think this would be a great place to pause before we move on to some of the molecular work that we're doing. And I'd be happy to answer any questions that are related to the algae program and how we developed our Algal Index in California.
Thank you, Susie. That was wonderful. I thought you did a really great job stepping us so clearly through the process you went through to compare and select the best metrics for the state. While folks, to give folks time to type in their questions, why don't I ask the first one, if you could, you mentioned using presence absence data in your O over E models. And I was curious whether you think that may have affected the performance of O over E models, observed over expected models compared to the multimetric indices.
Yeah, so I should clarify, we converted all of our taxonomy data to presence absence for both our O over E and our multimetric indices. And while it's common to do that for O over E index development, it is not common to do that necessarily for multimetric index development. We do think that it could have presented a challenge, and there is really important ecological information that is retained in relative abundance data, and it would be really useful, and sometimes it gives you important biological signal. For our purposes, we knew that we wanted to design this index to eventually be able to be calculated with DNA-based data. And so we wanted to really challenge ourselves to develop the index using presence absence data in the hopes that eventually it would be more compatible with DNA-based data, which we know has relative abundance issues. And generating quantitative data with DNA-based methods can be a little bit more challenging. So we wanted to design the original Algal Index with the eye towards the future, that we one day would be calculating it with DNA-based data.
Great, thank you. Here's a question from Mark. How was the diatom taxonomic harmonization managed? And what happened to the Southern California Diatom website?
Oh, that's a great question. Yeah, so the first question was about harmonization. And so we have, so thankfully all the data was collected using our standardized protocols for the state of California. So that was one common denominator that we have. We did perform essentially an indicator species analysis, but with taxonomists for our data set. So we were looking for evidence of a taxonomist signal in our taxonomy data, and thankfully we didn't find one. So that was good. In particular, part of that is the credit to the fact that we have a limited number of taxonomists who are doing the majority of the taxonomic data generation. And so there was a lot of consistency in that way. We have a harmonization process in California that's part of our Algol program. And so on an annual basis, the data is harmonized. And one of the other things that we did was we created an STE, a standardized taxonomic effort, essentially, that's now in use, and the taxonomists are using. And in the creation of the index, we essentially harmonized names so that if something was a morphospecies, it was dropped to genus level. And there were certain other ways that we tried to condense a number of unknown taxa that weren't officially described and that we were worried would be weakening the performance of the index because they were potentially two very similar species that were given two different morphospecies names. So there was a fair number of name harmonization that we did that way also. And then the second question, what happened to the Diatom website? So we do actually, Mark, if you head to, maybe shoot me an email, I send you the link. We now have an internal version of the website. It had been hosted by University of Colorado for way too long. They were doing us a big favor. So now we've recreated the index or recreated the website on the Scorp servers. So if you shoot me an email, I'll send you that link.
Okay, here's a question from Joe Mohan. You mentioned biovolume of diatoms and soft algae a few times. What method did you follow to determine biovolume?
Yeah, so for the diatoms, they're doing counts, valve counts. And for the soft algae, we have biovolume that's determined volumetrically by water displacement. And then the microalgal fraction is analyzed, and the macroalgal fraction, the relative abundances or the proportion of the different macroalgae is determined. And then it is converted using the biovolume estimates through displacement.
All right, and one more question before going to the next part of the webinar from Jan Stevenson. How widely is the Algal Index being adopted for use across the state, and how are stakeholders responding to its availability?
Yeah, great question. So the ink is barely dry on this index. So as you can see here, the manuscript is in press right now. So I think things will really accelerate when the manuscript is officially out, and the index is being calculated by a number of our user communities. So that includes both the statewide monitoring programs, some of our regional monitoring programs, but as you mentioned, the stakeholder community wants to begin taking some of their taxonomic data and running it through this calculator. So right now we have dreams and visions of how this index is going to be incorporated into a number of different programs, including, you know, one day in the development of biocriteria, in permit compliance, in 303D listings, but it hasn't gotten there just yet. I think it is still brand new, but we're excited to see it in the hands of the user and stakeholder community so they can really begin test driving it and hopefully writing it into some regulatory management decision-making.
Okay, great. Just for the sake of time, let's move on to Rafi, and he also has a comment to add.
Sure, can you guys hear me?
Yeah.
Yes.
Okay, I just wanted to comment on stakeholder reception. There is actually, I would say, a fair amount of enthusiasm for this. I mean, there's some stakeholders that think, okay, this is just another thing to worry about. But there are a number of folks, particularly in urban areas, where we're dealing with streams that are so pervasively altered that we have pretty clear evidence that we are not going to achieve reference-like scores for a bug index without massive, removal of concrete and other kinds of rehabilitation. And there's sort of hope to look for another way of assessing ecological condition that might be more responsive to management. And there's some evidence to think that the algae indices may provide a better way of evaluating, the impacts of their management when we know that the bugs are already telling us that everything's awful. So I would say there's actually a good amount of receptiveness for an algal index in at least certain portions of the stakeholder community.
Yeah, that's great. Okay, should I keep going? Ready to go?
Yes. Go
All
ahead.
right, awesome. So then for the second part of this talk, I really wanted to go over some of the ways that we are now exploring adapting this index for use with DNA or molecular data. So as I mentioned, we have this brand new shiny biological index that was created using morphology-based data, but we're already asking ourselves the question, can we calculate this index with DNA-based data? And I think, the Diatom Web Academy had a great presentation a couple of months ago going over some of the different ways that we're using DNA metabarcoding or DNA sequencing for index development. And so that was a really great introduction for some of the work that we're also doing here in California. So state agencies have now expressed an interest in exploring a DNA-based approach for generating the taxonomy data for biomonitoring and bioassessment applications. And so it really has the potential to complement morphological analyses. And in doing so, hopefully alleviate some of the capacity limitations that we face. As I mentioned, we have a minimal number of taxonomists that have been processing a number of our algae samples. They are overworked, and so we want to present an alternative to hopefully be able to complement the work that they're already doing. We also are interested in seeing if by using DNA-based approaches, we can potentially expand the suite of indicator species that we use for our bioassessment application. So as I mentioned, currently we're looking at diatoms and soft-bodied algae, but there's really potential now to also be looking at our microbial community and some of the prokaryotic species that are present in these environments, or other organisms that we haven't been able to identify previously because of difficult morphology or cryptic species complexes. So in this way, DNA-based approaches might be able to help us improve some of the biological tools that we have at our disposal. So for a quick reminder on what exactly is DNA metabarcode sequencing. So DNA metabarcode sequencing is our way to identify multiple different types of organisms all present in a single individual sample. And we do that by something called high throughput sequencing. So we start off with our environmental sample, and that can be a rock scrape, it can be a water sample, it can be mud, and it can include organisms or the organism DNA from a diversity of different types of organisms, microbial to charismatic megafauna, all ends up in our environmental sample. We take those environmental samples back to the lab, we extract the DNA, and then we amplify what we call DNA barcode regions. So these are really the snippets of DNA that we use for fingerprinting and identifying all the different taxa that are present in our samples. We then take our raw sequence data and subject it to bioinformatic processing, where we're cleaning out any erroneous sequences, we're getting rid of some of our junk data, and we're making sure we end up with a data set that is clean, pristine, and ready for taxonomic assignment. To do taxonomic assignment, we're really relying on our global DNA reference databases. So we take our unknown DNA sequences and we compare them to these known reference sequences that are available, and we begin to determine what species we have present in our sample. Like we do with our morphology-based data, we then can go forward and calculate our Algal Index scores. And so what we're really interested in evaluating here is how DNA data is performing in comparison to our morphology-based data in the eventual assessment outcomes based on these index scores. One of the roadblocks that we run into, though, in initially adapting some of these DNA-based methods for bioassessment is that there's really a lack of standardized protocols for how we should go about generating this taxonomy data for algae. So just to give you a brief overview, as we look throughout this process of collecting and then analyzing our DNA samples, there's multiple points when we have to make a decision about how we're collecting a sample, how we're preserving it, how we're extracting the DNA, what DNA barcode primers we're using, what bioinformatic pipelines we might be using, what reference sequences are available, are my taxa present in these global reference databases, and are our outcomes going to be the same? So I'll tell you a little bit today about some of the pilot studies that we're working on to address two of these key issues. In particular, getting at the question of what DNA barcodes should we be using for evaluating our algal communities, and how our assessment outcomes going to change or not with this DNA-based data compared to our traditional morphology-based data. So as a bit of an overview of how we performed this pilot study, we started with that algae composite sample that I mentioned we generate at the end of our sampling out in the field. we have our algae sample, we split it off into our morphology sample, and then we filter one part of that sample down for our DNA-based analyses. Traditionally, we then perform our microscopy and our taxonomic analyses, calculate our Algal Index scores. Now we're also adding a parallel analysis where we're taking our DNA sample, extracting the DNA, sequencing it using a number of these different DNA barcode regions, performing our taxonomic assignments, and then calculating our Algal Index scores. So as I mentioned, the big question we're trying to answer is how different DNA barcodes influence our eventual algal community composition and our index score performance. For a little bit of background on which different DNA barcodes we were interested in comparing, I'll give you just a brief overview. So we were looking at, we wanted to make sure that we could capture all of the different assemblages that we look at with morphology. So that means we're looking at the cyanobacteria and our eukaryotic algae, including our soft-bodied algae and our diatoms. So that means we had to use quite a few different DNA barcodes that we wanted to test drive. So first off, we targeted something called the 16S Rrna genes, and this is what we would use for getting a better sense of our cyanobacterial communities. But in fact, the primers that we used are universal to all bacteria and archaea. So we used some universal 16S Rrna genes, and then we also used some universal 18S Rrna genes. And so the 18S genes, as I mentioned, lets us look at our eukaryotic algae. So this will include your diatoms and your soft-bodied algae. But like I said, it's a universal eukaryotic primer, so we're really capturing any and all eukaryotes that are present in our sample. We also then looked at a barcode region that targets the RBCL gene, which is plastid encoded. It codes for a subunit of the Rubisco gene. And the gene, or the barcode primer set that we used for this comparison was actually diatom specific. So for this primer set, we're really only going to be zooming in on our diatom populations. To jump right to some of the preliminary results that we've observed with this analysis, as you might expect, using your DNA barcode primer really does make a difference. And so we saw really big differences in the community composition of our samples, depending on whether they were sequenced with each of our different individual DNA barcodes. So this NMDS plot that I'm showing you here is an ordination of all of the different diatom species that we identified as an example. And so I've color-coded all the different samples according to which of the different analytical techniques I used for generating our diatom species lists. So you can see in green, those are the species lists that were sourced from doing our DNA barcode sequencing using the RBCL primer. We have the two different 18S primer sets that we were comparing. And then we also have a comparison of the morphology-based diatom community composition. We can see based on this ordination, there's really big separation with the different community composition from same exact samples, depending on which of the different DNA barcode regions we were sequencing. So while we were targeting, for example, the diatom populations with the 18S primers, it's the same exact gene, but with two different barcode regions that we were sequencing, and we still see really big differences in those populations. So the barcodes that you choose really does matter. And that influences your resulting algal index score. So the figure here on your right is showing you, again, the 4 different permutations of how we were generating diatom community composition data on the x-axis, and then on the y-axis, we have our diatom index scores. You can see that in general, our morphology scores were higher than our DNA-based algal index scores. What we can see is that our diatom-specific barcodes, here colored in green on this box plot, in general outperformed some of our other DNA barcodes that we were test driving. They tended to be most sensitive to our stressor gradients and the most similar to the morphology data. So for these analyses where we're using morphology data as a bit of a benchmark, although we were open to the idea that we expect fairly different community composition to arise from DNA-based analyses in comparison to our morphology data. But we did in general see that our RBCL data tended to trend more similarly to the algal index scores that we were seeing with our morphology-based data. But again, you can see based on these box plots, there's just some really big differences based on index scores that were generated with some of our other 18S barcode primer sets. To look at some of this information in a slightly different way, I'm showing you a series of Venn diagrams where we're trying to get a sense of what species were we picking up with morphology versus DNA, where was their overlap, and where were we missing species with either of these two methods. By and large, what you can see is whether we were targeting our cyanobacteria or our diatom populations or our soft-bodied algae. And if we were looking at individual species composition or even zooming out to a higher level and looking at what genera we were identifying, across the board, we were just missing a ton of the taxa that we expected to see based on our morphological analyses. So while we were sometimes picking up novel taxa that we otherwise hadn't identified with microscopy, there were just across the board with all of our different barcodes, a number of species that we were missing. And the main reason that we were missing these taxa is because they lack DNA reference sequences in our DNA reference libraries. So this pie chart that you see on the right-hand side of your screen is just a quick visualization of how many of our taxa that we expect to see in streams in California, we know we'll be able to see using DNA-based approaches. So for this analysis, we looked at our master taxa lists for streams in California, which have about 1,700 different species on them. And we were getting a sense of whether or not those species have reference sequences available in these global DNA repositories. And about 60% of our taxa don't currently have reference sequences available in these repositories. This lack of DNA reference sequences, and likewise a lack of trait attributes for a number of these key taxa that are important for the calculation of these algal indices, is really limiting the index performance. So those low scores that we were seeing with the index performance was attributed to the fact that we are just essentially missing, or you go blind to some of the taxa that you can pick up with DNA-based analyses because they're either lacking reference sequences or they lack necessary trait attribute data for calculating our index scores. But it's not all doom and gloom. We have a plan. And so there are future options for how we can go about adapting our algal index so that it hopefully performs better with DNA-based data. So first off, we think about how we can update the existing algal index. So one way of doing that is looking at our metrics and seeing which of them, by and large, were performing poorly because of lack of trait attribute data for the taxa that we pick up with DNA. So we know that there are some novel species that we're picking up with our DNA-based data that currently don't have some of that autoecological information that we already have for our morphology-based species. And so can we just drop some of those metrics that by and large are biased because we lack trait attribute information for those key taxa? The other way to address that need would be to generate trait attribute information for some of the abundant taxa that we're seeing with our DNA-based analyses. So that's another way that we can begin to close that gap between taxa that we're picking up with DNA and those that have available trait attribute information. One of the other things that we could do that we'll be exploring is grading on a curve. So can we adjust our algal index scores when we're using our DNA-based taxonomy if there is in fact a consistent bias in our algal index score? So far we've seen that by and large our DNA-based scores are depressed in comparison to our morphology-based scores. If we can show that that lower index score is consistent for DNA-based data, we can essentially just grade our scores on a curve. And if you generate your data using DNA-based methods, it's just corrected so that it is can be compared to morphology-based data as well. Our third option is exploring something called a taxonomy-free index. And I know you've heard a little bit about this in a previous webinar, but the strengths of a taxonomy-free index is we're no longer relying on whether or not a DNA sequence has an available reference sequence to match to in these global DNA database repositories. Instead, you begin to assign trait attribute information using the occurrence of those DNA sequences across different stressor gradients. You're essentially developing your own trait-based attributions based on the stressor gradients that you observe in your own samples, and therefore you're not reliant on the previous trait attribute information for your different taxa. The taxonomy-free index requires a large data set with a high number of reference sites, same as when we developed our algal stream condition index. We want to make sure that we're calibrating it on a robust data set with strong representation in our reference site pool. And so getting there means having, a couple hundred DNA sequences from across the state. So it's a challenging analytical effort, but one that I think we're poised to do. We've been collecting DNA samples across the state now for 4 years. We have over 600 DNA samples so far. And so we're really positioned to be able to explore the development of a taxonomy-free index. But one thing that we're also leaving the door open for is that adjusting the Algal Index to perform with DNA-based data might be some combination of all three of these approaches. And so we're going to be exploring each of them to see what's going to get us closer to being able to calculate strong, robust, sensitive, accurate, precise Algal Index scores using DNA-based data. So with that in mind, there are a couple of things that we're also thinking about to make sure that we're positioned to be prepared for this eventual DNA data deluge, really. So I think one of the things that is sometimes intimidating for novice DNA users and some of our stakeholder and regulatory communities, if you haven't worked with DNA data before, sometimes you don't know where to begin, and the influx of data can seem really overwhelming. As we know, DNA-based methods can help us identify between 10 and sometimes 1,000 as many species as we're identifying with traditional methods. But that means that you're generating gigabytes and gigabytes and gigabases of data from your DNA sequencing runs. So what we're thinking about is how to prioritize making this data useful and actionable for our user communities, and especially addressing that need of having standardized approaches for how we're handling this deluge of data. So one of the ways that we're focusing on making sure that our data is comparable and of high quality, no matter who's generating it and who's analyzing it, is through the work of the California Molecular Methods Work Group. So this was a work group that we founded in 2018. It is a work group of the Water Quality Monitoring Council here in California. And it's comprised of a number of different technical experts and then also representatives from our stakeholder community. So the distribution of icons that you see on the right-hand side of your screen just gives you a sense of the diversity of the members of this work group that we have working together now to develop standardized approaches for collecting, analyzing, and reporting molecular data. But really this work group is to serve as a clearinghouse for these molecular methods and also as an opportunity to enhance collaboration and communication among not only the scientists on the ground or the technical experts who are used to using this data, but those that are going to be implementing this data for management and regulatory applications. So hopefully by getting out ahead, we're developing these standardized protocols so we can ensure that our data is going to be comparable and of high quality no matter who's using it and collecting it, analyzing it anywhere throughout the state when we really start to get going with more common collection and analysis of these DNA-based approaches to bioassessment and biomonitoring. One of the other ways that we're thinking about making sure that all of the data that we're collecting is getting turned into something useful is investing in the development of visualization tools. So SQUIRP has been pioneering, I would say, some of these really interactive online tools where we can take our bioassessment data and make it useful for our management communities. This is an example of one that we've developed for the Stream Quality Index dashboard. It's useful here in Southern California. And as you can see, you can head to this website, you can zoom around, find some of your favorite sampling locations, and if you click on those individual sites, it'll give you a distribution of different biological index scores for that site, the comparison of those index scores to reference site scores in the area, what some of the potential causal stressors might be for those individual sites, and it will help you evaluate what's causing some of your lower index scores or what some of the stressors might be that are causing the different distribution patterns or responses to implementation changes that you've been making. And so it's a really great way for our user community to explore their data. And this is what we're visualizing happening for DNA-based data as well. And we know that behind the scenes, a lot has to happen when you collect a sample and turn it into DNA taxonomy data, but we want to make it as user-friendly as possible for our management community. And we really think that these online and interactive tools are one of the great ways that we can begin to do that, to make sure all the data we're collecting is getting turned into something that is actionable. So lastly, I just want to end by saying, you know, I think in this group, we're all fans of algae in general, and hopefully also in bioassessment. And we really see that we can develop these powerful tools that will help us generate better data, which helps us make better decisions and leads to better outcomes. And I think the standardization that we're focused on here and the sophisticated analytical techniques that we've been working on developing are really going to help us realize the potential of these tools for broad application. And so I'll end here with just a couple links. As I showed before, there's a link to learn more about our Algal Stream Condition Index if you're interested. And also, if you want to learn more about the activities of the Molecular Methods Work Group, you can head to the link here. But otherwise, thank you so much for your attention. I know this was a lot, but it's really fun to share all of the work that we're doing in California with all of you. So thank you.
Thank you, Susie. You went over a lot of great information, and we are over time. But I'm happy to see that most of our participants have stayed on. I think it shows the timeliness and interest in the topics you covered. For those who have to leave, I'll make sure to send out a link to the recording. And you can also send your questions by email to the Diatom TCC email address or to our speaker, Susie. And for those who can stay on for a little bit longer, we'll address a few questions. There was one from Julianne Heinlein before the break. She asked, do you find that the hybrid MMI gives enough added benefit to justify the species level soft algae counts for these kind of broader general bioassessment questions?
Yeah, that is a great question, and it is extremely timely. So we are definitely spending a lot of time thinking about the value add of collecting the additional species data. Our hybrid multimetric index has excellent performance, but so does the diatom only index. And so right now we don't have strong differences in the hybrid versus the diatom index. So while the soft-bodied algae are helping the hybrid index perform well, if we didn't have access to soft-bodied algae taxonomy, we thankfully still have a high-performing diatom multimetric index. Now, one of the things that we think could help enhance some of the performance and help us, I guess, capitalize on the biological signal of the soft-bodied algae community is beginning to explore these DNA-based methods. So I think that it's not the shortcomings of the soft-bodied algae population, but I think we're limited in some of the taxonomic data that we have accessibility to now. But with the advent of the DNA-based methods, I think we can see some new tools develop where we're harnessing the biological signal of those soft-bodied algae communities even more effectively. So I think it's really going to be a decision for our program managers if they are deciding to move forward with our diatom multimetric index or the hybrid index. And so I'll be interested to see what the program managers decide to do coming down the line. But I'm also optimistic that right now, if they decide to move forward with a diatom only index, in the future, we might begin to see the incorporation of the soft-bodied algae index once we move to DNA-based methods that just make it a little bit more accessible.
All right, here is a question from Melissa. Were you able to approximate relative abundance of species using the DNA-based analyses, or were these only looking at presence absence?
Yeah, great question. So because, as I mentioned, we constructed our index to be presence absence only, that's how we convert our DNA data right now, is to presence absence only for calculating our index scores. What we're seeing from a number of our colleagues and collaborators and some of the efforts over in Europe for development of DNA-based diatom indices is that there's some ability to reconstruct the relative abundance by incorporating an element of species biovolume and using that to correct your read counts for your different DNA sequences. So some of your larger taxa, you can correct the numbers of read counts in comparison to some of your smaller taxa. And so it's really on the vanguard, I think, of how we're using DNA-based data for calculating algal indices, but there's real potential to be able to control for relative abundance numbers in your DNA sequence-based data. So I think that's something we'll see more of moving forward. But so far for our index in California, because we use the presence absence data, that's how we treat our DNA-based data.
There's a question from Lawrence. Did you find any interesting species using morphology and molecular methods?
Interesting. I'd say there's an argument that all of our algae are interesting in some way or another. I think maybe the question is about who we're picking up differently. And there were large differences between who we pick up with DNA-based approaches versus our morphology-based approaches. I think we're going to be spending a lot of time really evaluating where the key differences are and returning to some of those taxa and saying, okay, if there were ones that we missed with DNA-based methods because we don't have reference sequences available for them now, can we go about generating those reference sequences ourselves? And so that's something that we've partnered with some of our taxonomic experts to do. And so over the next 3 years, as we begin to develop this Algal Index, we have a parallel effort to hopefully also bolster those DNA reference libraries for the key texts that we were missing that we know should be there based on the morphology data.
Well, I think we should wrap up here. And I want to thank again Susie and Rafi for developing this webinar. It was very interesting. And I thank everyone for joining us today. I look forward to seeing you all in a couple weeks. It will be a taxonomy focused talk on the genus Eunotia by Dr. Paula Fury. So please join us then, and thanks again, everyone. See you.
Great, thank you.